Well, hello, my name is Rodolfo Pelinson. I'm going to present you part of my PhD research, which is titled Assembly and Reassembly of Freshwater Communities Under the Influence of Agrochemicals in Different Spatial Contexts. So, if you walk into a pasture, something that you often see is these kinds of habitats. These are temporary ponds and they are usually filled with rainwater during the rainy season. And if you walk into a sugarcane field, you will see very similar environments. And these environments, they harbor a great diversity of organisms. And here our focus will be on aquatic insects and amphibians. And these crop fields, they also are very often treated with different pesticides and fertilizers. So something that we asked ourselves was, how does the use of these agrochemicals can affect these communities? So this is not really a new question. Some other works have investigated the effects of different dosages of pesticides on species richness, survival, and species interactions. But something that these studies have ignored is, first, they lack a more realistic use of agrochemicals within a crop cycle, a real crop cycle. And two, they lack spatial context. So we believe that the meta-community framework would be an ideal approach to understanding how these communities can be affected by agrochemicals. So using this framework, we can put together these three high-level processes of community ecology: niche selection, ecological drift, and dispersal. And here in our approach, niche selection is actually the use of different agrochemicals. They actually represent different selective pressures. And the dispersal processes will be represented by different spatial contexts. So all of these processes, together with the ecological drift, of course, will all be affecting community structure. So first of all, I want to talk about how dispersal processes may affect these communities. So in the case of aquatic insects, we know that some of the herbivores and detritivores, like mosquitoes, they have smaller body sizes, so they cannot disperse very far. However, they do have larger population sizes and shorter generation times. This makes them to have many events of dispersal, so they can actually easily colonize very isolated habitats. Their predators, however, they do have larger body size, so one individual of a predatory insect can actually reach a highly isolated habitat, but they have smaller population sizes and longer generation times. So they have very fewer events of dispersal when compared to their prey, which makes it difficult for them to successfully colonize very isolated habitats. So what we actually see in the real world is a gradient of decrease in the abundance of predatory insects, and this decrease in abundance have a cascade effect to the lower trophic level, and what we actually see in the lower levels is a opposite gradient, an increase in the abundance of herbivores and detritivores. So these effects of dispersal, they should affect the selective pressures that we are working with here. So for example, fertilizers, they should increase primary productivity. So when we have an increase in nutrients, we should observe an increase in the biomass of algae. And in lower isolated ponds, what we should see is that increasing resources to actually affect the abundance of the higher trophic levels, like the predatory insects. However, when we go to a higher isolation, we do see dispersal limitation of predatory insects, and we should observe an increase in the abundance only of herbivores and detritivores, and not so much of these predators. So, and when we use herbicides, what we should actually see is, if the addition of nutrients should increase algae biomass, the use of a herbicide which is specifically manufactured to have negative effects on plants, it should actually have the opposite effect and counteract the effects of nutrient addition. And when we talk about insecticides, well, insecticides, they are specifically manufactured to have negative effects on insects, so they should reduce the abundance of aquatic insects. That in turn should have an indirect positive effect on amphibians, because their predators and competitors they had its abundances reduced. And as we know, insecticides are applied in pulses, so communities should recover after some time after the application. And this recovery should be faster in lower isolated communities. So what we expect what you're expecting here is an interaction between spatial isolation and these different selective pressures. So to test this hypothesis, we made an experiment, a field experiment, where we had here in blue a stream, and the floodplain of this stream working as a source of insect and amphibian colonizers, and we constructed a row of artificial ponds at 30 meters of this source of colonizers. This was our low isolation treatment, and we constructed other two isolation treatments, 120 meters and 480 meters from this source of colonizers, and the distance between these ponds were of 30 meters. So a third of these ponds we treated as control, so we did not add any agrochemicals to them. They should represent native habitats. Another third we treated with fertilizers, and these ponds, they should represent ponds that are embedded in a pasture land of medium intensity, so it's a pasture that actually uses fertilizers. And the other third, it's supposed to simulate a pond that is embedded within a sugarcane field. So they were treated with fertilizers, a herbicide 2,4-D, which is the second top seller in the state of São Paulo, and an insecticide, which is the top seller of the state of São Paulo. And we adjusted the dosages per hectare of all of these agrochemicals to the area of our ponds, which was 4 square meters. This is an example of one of these ponds, and they were all filled with 1,200 liters of water, of well water actually. So we try to simulate the most common sugarcane crop cycles. So we have here our rainy season, which goes from September to March in southeast Brazil. And by the beginning of the rainy season, we filled our ponds with well water. And in October is when the sugarcane harvest usually happens. So this was when we made a first pulse of insecticide and started our weekly additions of fertilizers. And 1 month later, it is when the post-emergence herbicide is applied. So we made a pulse of the 2,4-D herbicide. And along this time, we made 3 surveys of the aquatic communities, insects and amphibians. So this is our results for the 3 surveys separately. As you can see, we did see main effects of land use and isolation, but we did not see for any of our surveys that interaction effect between isolation and land use that we were expecting. So I'm going to present you results for isolation and land use separately. First, talking about isolation, as we can see, we did see differences between lower isolated ponds and highly isolated ponds in all surveys. Here I'm showing only control ponds just for simplicity and to improve visualization, but this effect repeated itself across all treatments. And we can see that for this first survey, the negative effect of the isolation on predators' abundances were only negative for one species, so we had a weak effect. However, herbivores and detritivores did have were importantly positively affected by isolation, and predators still had a idiosyncratic response to higher isolation treatments. In other surveys, and herbivores and detritivores still had this somewhat positive effect. By the end of the rainy season, all species were being negatively affected by isolation, probably because it was already the end of the rainy season and the abundances of almost all species were declining. Now comparing pasture ponds to native habitats, we did not see these are control ponds and these are pasture ponds. We did not see any differences between control and pasture ponds. So we asked ourselves why. So we looked at nutrient concentrations of these two treatments across the experiment. This is total nitrogen at 74 days after beginning of application, and this is total phosphorus 125 days after the beginning of application. And as you can see, there is no differences between nutrient concentration in both these ponds. And I'm showing here in this hashed line the threshold to which a pond is considered hypertrophic. So as you can see, even our native habitat ponds are considered hypertrophic. So we don't believe now that nutrients may be a limiting factor in these habitats. Even habitats that don't receive fertilizers do have a lot of nutrients. So now comparing sugarcane ponds to native habitat ponds or control ponds. The first thing that we see here, I just changed the days here. Now we are looking at the surveys 4 days after the application of insecticides, 40 days after, and 120 days after. So we see a striking difference between sugarcane ponds and control ponds in all surveys that we did. And when we looked at what species were driving and what patterns were driving these differences, we saw that indeed the abundance of aquatic insects were highly reduced by the application of the insecticide, and that effect kind of fades away as the end of the rainy season approaches. And when we looked at amphibians, they did not respond to the application of insecticide right after that application, but a few days later, actually 40 days later, they did increase in abundance, probably as a consequence of the decrease in abundance of their predators. So before we summarize our results, something that is important to say is that here we only looked at one dimension of the land use intensification problem, which is the effect of agrochemicals on the community structure, on differences in abundances patterns. So there are other important effects of land use or even the use of agrochemicals. For instance, we can look at behavioral and physiological changes that could be induced by pesticides. We can look at differences in dispersal limitation in real crop fields and pasture lands compared to native habitats. We can look at differences in environmental heterogeneity within and between ponds in different land use types. So the aim of this study was only to look at the effect of agrochemicals on community structure. And regarding that, we did not see many important differences between pasture ponds and control ponds. However, when we compared sugarcane ponds to control ponds, we did notice that sugarcane ponds might be experiencing an important decline in the abundance of aquatic insects, while amphibians may be increasing in abundance in those ponds. And something that is important is that this effect were not dependent on spatial isolation by distance. However, we still don't know what would be the effect of a real crop field matrix on dispersal limitation of these species. So here we showed the importance of considering the effects of land use on community structure in more realistic scenarios, as some theoretical predictions may not always be fully realized in many real-world conditions. So this is my email for contact if you are interested. And thank you for all the people, all the institutions, and all the funding agencies that supported this work. And thank you for watching this video.